It's biology with Mr. B. Biology with Mr. B. That's me! Right, hello everybody, it is Mr. B here. And in this revision video, I want to go through ideas about cell division and stem cells. So it's just not going to be possible for me to go through absolutely every single little bit. That'll be absolutely silly in the time limit I've got. So instead, I'm going to focus on what has come up more than others so far in the sort of couple years of exams that we've had. So I'm going to make sure I go through the cell cycle. I'm going to go through embryonic and adult stem cells, what the differences are. I'll look at therapeutic cloning a little bit. That's a nice grade 9 concept. And then, if time permits, I will try and get through the swift ideas about cancer. So for meristematic uses, which are stem cells in plants and how they're used commercially, please make sure you're looking at the spec points. For OCR, the spec points are really, really clear with what information the exam boards wants. Okay, cool, let's roll. So I want to start with the cell cycle. The cell cycle is effectively trying to just explain what a cell is doing during any part of its lifetime. And we can split it into a pie chart with 3 steps. A very large step 1, and then much smaller steps 2 and 3. So again, think of this like a cell's life cycle. It starts here, it spends a lot of time in stage 1, and eventually moves into stage 2, and then stage 3, where we then back again to stage 1. And a cell will constantly be doing this throughout its cycle. And effectively, what we need to think of it is the cell cycle is the constant process of a cell dividing by mitosis. So one cell cycle should mean I get one division of the cell by mitosis. And here's what I know if this is a mitotic division, mitosis-based cell cycle. I know I should be producing 2 genetically identical daughter cells at the end of this process. So not only do I want my cell to divide, I want the 2 new cells produced to be genetically identical. If the parent cell was 46 chromosomes, I want my new daughter cells to have 46 chromosomes as well. And ideally, for these cells to function, I want them a reasonable size and shape similar to the parent cell. I also want a reasonable number of cell organelles. So, if this cell had, say, like 5 mitochondria, I'd want these cells to have roughly 5 mitochondria for them to, function as the potentially specialised cells that they may be. Okay, so step 1 of the cell cycle. Now it's not essential that you learn this term, but step 1 is known as interphase. And during interphase there are 3 things happening to prepare the cell for division. So interphase, this really long step, prepares the cell for division. And again, if I remind you, I want my parent cell with 46 chromosomes, if it's a human being. I want it to divide, and I want it to be roughly the same size afterwards. I want it to be genetically identical. And I also want the same sort of number of organelles like mitochondria. So clearly, think about this for a second. If this cell just divided in 2, so boom, down the middle, you're going to end up with two cells that are half the size. If the nucleus just divided down the middle, you're going to end up with 2 cells. One is 23 chromosomes, the other is 23 chromosomes. That's not efficient, that's not correct. And we're also going to split up all the organelles. We're only going to get half the number of mitochondria, half the number of ribosomes. So in this step 1, we need to fix that. We need to make sure that the nucleus, when it divides, both of these cells get 46 chromosomes. The only way you can do that is to actually duplicate all the genetic information in this nucleus here first. So during interphase, one of the main steps is that we actually duplicate, we can call it replicate, the DNA. It means there's twice as much genetic information. Please don't say the DNA doubles, that's not true. Technically you always have 46 chromosomes, just each chromosome now has just extra DNA, replicated DNA. So clearly these cell organelles, I've got 3 this side, yet 6 that side, so clearly I need to make more. So I'd make more mitochondria in this cell before it divides. And the sizes, I want them to be roughly the same size, so this one needs to grow. So put those 3 steps together. If these 3 things all happen, then the parent cell will be ready to divide. So interphase, step 1, DNA's got to duplicate. You've got to increase the number of cell organelles, like mitochondria and ribosomes, and the cell has to grow. If these 3 things happen, then the cell will be ready to divide, and when it does so, the 2 new cells produced will be genetically identical, because you duplicated the DNA, so when it splits in 2, you've still got your 46 chromosomes. They'll have enough organelles to carry out their function, and they'll be large enough in the correct shape and size to carry out this potentially specialised function as a cell. Okay, step 2. So the advantage of these videos, guys, if I am going a bit too fast, pause me. So step 2 is a term you do need to know. Step 2 is where mitosis actually happens. So mitosis is not the final step. When mitosis finishes, you won't be left with 2 genetically identical cells. Mitosis is a term we use for when the nucleus divides. And how it does that is that the chromosomes in the centre of the nucleus get pulled apart to opposite sides of the cell. So if I try and draw a cell here, so the nucleus. Now we don't need to know actually how this happens, but I'm just going to do a little bridge to A level just to give you an idea. The nucleus actually breaks down, and it means that all the chromosomes in their duplicated fashions, hence why each one's got a couple lines, they actually line up in the middle of the cell like this. And then a slight contraction happens and they get split, where half go this way to the right and half go to the left. Half go to either pole. And so we end up with a cell that's got half its chromosomes this side, half its chromosomes that side. But because they were duplicated, actually each side now has 46 chromosomes. A brand new nuclear membrane forms, and we're left with a cell that has 2 nuclei. That's the end of mitosis. The nucleus has divided. So just a link back to cell structure. In cell structure, we learned that prokaryotes don't have a nucleus, which means prokaryotes can't do mitosis, as they have no nucleus. So when prokaryotes divide to make genetically identical daughter cells, daughter bacterias, they divide by a process called binary fission. So prokaryotes do binary fission instead. Okay, so we're now in a situation where we have a cell that is, twice as large as it usually is, thanks to interphase. It's got twice as many cell organelles thanks to Interphase. It's because of mitosis, it now has 2 nuclei, each with 46 chromosomes, if this was a human. So the third stage is to actually get 2 genetically identical cells out of this. So we do not need to remember this term, but step 3 is called cytokinesis. And in cytokinesis, if I redraw that cell with the 2 nuclei, and I'll just draw a few mitochondria in there as little sausage shapes. I'll draw a few ribosomes scattered around as well. Do like me a ribosome. Site of protein synthesis after all, very important. So, this final step, I want you to imagine a pair of scissors literally going snip, snip, snip and snipping down the middle of this cell. So which parts of the cell has it actually snipped? Which parts of the cell has, hi cat, which parts of the cell has the line actually gone through? It's gone through the cell membrane and it's gone through the cytoplasm. I appreciate my line has also gone through a tiny part of my mitochondria there, but I promise you, the mitochondria would just move out of the way. So for cytokinesis to get the credit in the marks, you need to state that the cell membrane and cytoplasm divide. So both divide. That's cytokinesis. And you are therefore left in 2 genetically identical daughter cells. And that is the cell cycle. Right. Next job then. So I'd like next to focus on ideas about stem cells. Just looking at the timings, guys, it's going to be unlikely that I get towards the end of what I planned. So I think the cancer topic, very much like the meristematic stem cells in plants, the spec points are fantastic for learning exactly what you want to say about like benign tumours, malignant tumours, how they differ, and so on. So please use those spec points instead for cancer, because I'm going to focus on my stem cells now, different types of stem cells. So a stem cell. A stem cell is an undifferentiated cell that has the potential to become many other cell types. If you hear that noise, that's one of my cats being absolutely stupid. Heyo. I think if the cat gets on camera, it will make this far more interesting though, so let's keep rolling. So a stem cell definition is really crucial, guys. It's a 2 marker. One for identifying it as an undifferentiated cell. That means it hasn't done the process of differentiation because it's not a specialized cell. But it also has the potential to become many other cell types. A nice example, stem cells in your bone marrow, so in the hollow in your bone, they have the ability to become bone cells, blood cells, and even skin cells. So those stem cells can become many other cell types. And if you really want to go into it, if you think about like embryonic stem cells, embryonic cells, well, they have the potential to become any type of stem cell. Which leads me to where most of the marks and the credit have come from so far. It's the appreciation of embryonic and adult stem cells. So what I'm going to do a quick table to run through the key sort of similarities and differences between these groups. Okay? So embryonic stem cells, they are of course found only in embryos. You can also get very similar stem cells from the umbilical cord as well that you can access. So found in embryos. Whereas adult stem cells are found, in all fairness, mostly all over your body, even as an adult. But the best example are the adult stem cells in your bone marrow. They're a really nice example to talk about. So embryonic stem cells, they are fantastic. They are fantastic and have fantastic potential because they can become, I'm actually going to think, I'm going to use the word differentiate because that's the correct terminology. They can differentiate into any type of cell. Whereas adult stem cells, they can differentiate into fewer cell types. So they're still good, but cells in my bone marrow can become like 3 or 4 different specialised cells, but they can't become everything. They can't become new nerve cells. They can't become new blood cells. Yes, they can. That's the bone marrow. They can't become new muscle tissue, for example. But the ones in the embryos can do. So this is the first big plus about embryonic stem cells from a scientist's point of view. I can use embryonic stem cells to treat any condition. Paralysis, type 1 diabetes, embryonic stem cells all the way. Adult stem cells, I can't. I can only treat the conditions to which those adult stem cells can actually produce cells. So I can use bone marrow to help with, you might know, bone marrow transplants to help like blood cancers and things, because obviously you can make brand new white blood cells with them, so great, but I can't use bone marrow to treat paralysis. I can't use it to grow new nerve tissue. I would need an embryonic stem cell. So, leaves us with some other ideas though. If you take an embryonic stem cell, there is a risk of damaging the embryo. There's a risk of damaging it so much that it could never become a human being. Some people see that as murder. And they're well entitled to that opinion, because at what stage does life become life? That is very much our own opinion. And if you damage that embryo, which means it can no longer grow and differentiate into an actual human being, it loses that opportunity, then obviously that is a significant ethical risk. And quite simply, embryos cannot give consent. If you compare it to adult stem cells and people taking your bone marrow, so don't get me wrong, the taking bone marrow is quite a painful process. But it's not life-threatening taking your adult stem cells. And obviously adults can give consent. We can choose to do it or not to do it. Okay. And finally, sorry, just laughing at my cat. So finally, embryonic stem cells and adult stem cells. If I can use stem cells from an adult, if I can use my own stem cells in particular, there will be no risk of rejection. But of course there is no possible way, at least naturally, for me to use my own embryonic stem cells. And therefore, if I use someone else's embryo, there is a risk of rejection due to different tissue types. So the final thing I'd like to leave you with is actually a grade 9 idea of therapeutic cloning. So again, grade 9 idea right now. therapeutic cloning. Embryonic stem cells are clearly, they are the best thing scientists get hold of if you're looking to cure and treat disease. How you do it though, in therapeutic cloning, is you also want to take into account that I can't just take a random embryo and expect it to work in my body. If they have a different tissue type, then my body will reject it. I could get an embryo, I could use it to create a brand new kidney maybe. The technology is there to become and create new organs, even if I use it just to create new skin cells. Say I had a burn, I was trying to fix my burn with a skin graft. If it's not my DNA in those cells, those tissues will have a different type, and it's likely my immune system will recognise them as foreign and attack them, which is what we know as rejection. But what if, this is Mr. Bateson, this is me, what if I was to take just one of my cells and take the nucleus and insert it into an empty egg cell? So this is an egg cell with a Mr. Bateson nucleus. And then I subjected that egg cell with my nucleus to a little bit, you actually add a little bit of electric current to it, a couple of other chemicals, and you generally will produce, as long as you can trigger it to start dividing, you can actually produce an embryo. With, in this case, Mr. Bateson nucleuses. That's me, but it's not me. That's my clone. And now, because these are embryonic stem cells, they can differentiate into any cell type. And because they've got my DNA, my body's not going to reject them. That's therapeutic cloning. And that is how you can also use adult stem cells to treat any condition. As long as you have a nucleus from the adult, if you can pop it into an embryonic stem cell and ensure that it's only got your DNA, nothing else, then you can start to use it as any other embryonic stem cell and differentiate it into any single cell type. So, thanks very much for your time, guys. I've gone 22 minutes this time, so that's not too shabby. So for those other aspects of the cell division topic, so for cancer and the meristematic tissue implants, the spec points are fantastic. And obviously those at King Edward's who have access to my revision booklet, please use that as well. For the other little ideas that were there about differentiation, I've kind of included what differentiation is and the general ideas about the genome and how things lie. That's something we can look at once again in the inheritance topic, but again, do check out my revision booklet to clarify that for you. Right, thanks for your time, guys. See you again really soon. Bye bye.